Alright, so we're going to start up now with our what should be last section on the overview of animal diversity. And the next feature that we're going to look at is segmentation. Alright, so segmentation is the idea that the body is built with repeating, well, segments or components that are somewhat redundant or somewhat repetitive. And so a couple of things that this does for animals that have evolved it is, number one, it does provide for some backup redundancy, so to speak, so that if part of the body is damaged, if other segments are essentially built the same way and do the same thing, the animal might survive because the other segments that are left can pick up the slack and kind of carry on. Um, Earthworms are a really good example of this. Earthworms, again, are in the phylum Annelida, so they're called annelids, along with leeches and one other group called the polychaetes. Really good example of segmentation. In fact, they are called the segmented worms because they exhibit this feature so predominantly. Um, the other advantage of segmentation is flexibility. And so the best way I have to kind of explain this or think about this is if you can imagine trying to put on a suit of armor that was made out of just one solid piece of steel. Like, even if you could get into it, it would be really, really difficult to move because you'd be restricted at your joints. You'd be restricted at your shoulders and your elbows and your wrists and your hips and your knees and your ankles and everything else, toes and fingers and everything, neck, right, your back. Everything would be very, very rigid because you'd be in one solid piece of steel. If you have segmentation at the joints, however, it makes you a lot more able to move. It gives you more flexibility, allows you to move around. And so it turns out that some of the biggest groups of animals are those that have evolved segmentation. So the arthropods, which again, I've said that name a few times now, but arthropods are things with jointed legs. So insects, spiders, ticks, mites, millipedes, centipedes, crustaceans, all of those are arthropods. Chordates, especially the vertebrates. So if we look at ourselves, we have segmentation in our backbone. Our vertebrae are segmented. Now, you could also, I think, make an argument that our limbs are segmented as well, but really, technically, the segmentation that we exhibit is in our backbone. And it shows up really well with those segmented vertebrae. And if you ever know anybody that's had vertebrae fused because of an injury, they lose a lot of mobility and they get a lot of restrictive motion in that part of their backbone because they've lost essentially a joint, and that makes it more difficult to move around. So we see a lot of segmentation in the really big groups of animals. We see it in the arthropods up there with that beetle up there on top. We see it with the earthworm, the picture down there on the bottom. So it's a really common feature in some of these animal groups that are especially diverse. So that kind of gives us a quick overview of some of the main features of animals that have evolved over time. So what we're going to do is go back and take a look at some classification in sort of a more modern contemporary way of looking at things. And we're also going to take a look at how taxonomists think the animals actually evolved as a group. So there were some competing hypotheses about how people thought animals came to be in the first place. And so we'll take a quick look at kind of the three of the most popular hypotheses, I guess you could say, and we'll talk about which one is the one that's been sort of decided upon as the correct one. So before we do that, we talked about our animal groups, modern day animal groups. The parazoans are those that don't have symmetry, don't have tissues of any kind, don't have organs. They're very, very simple, and really that's just the sponges. Don't need that in here. Hang on a second. Sorry about that. The other main group is the Eumetazoa, and those are the animals that do have symmetry and tissues, and they're protostomes or deuterostomes, and they have segmentation typically and those kinds of things. So basically the vast majority of animals are the Eumetazoans. The diploblastic animals within the Eumetazoa are the Cnidaria, so jellyfish and so forth, because they only have two germ layers as they develop, endo and ectoderm, but no mesoderm. And then triploblasts, which do have all three layers, endo, ecto, and mesoderm, again, is the vast majority of the animals, everybody except for the Cnidaria, and of course the Porifera, because they don't have any germ layers at all. So, going back to our original question, back in our first segment of the animal diversity lecture, the idea was that somebody brought us an animal that we didn't know what it was, and it was our job to classify it and try to figure out what group it belonged to. So this is kind of what we've looked at so far. This is kind of our summary of what we've seen so far. You open up the box and you find this animal that nobody's ever seen before. The very first thing you would look at is whether or not there are tissues. And if there are no tissues, your job is done. You know it's a sponge. It's a parazoan. If there are tissues, then you take a look at whether the animal has bilateral or radial symmetry. And if it's radial symmetry, we know it has to be diploblastic because the animals that are radially symmetrical are only diploblastic. Again, cnidarians are that way. If it has bilateral symmetry, only one line of symmetry you can draw down the middle, called the sagittal plane, then we know it's going to be a triploblastic animal and one of the many, many other groups of animals. Once we know we're dealing with a triploblastic bilaterally symmetrical animal, we would then take a look, for example, at whether it's a protostome or deuterostome. And in section 2 of the lecture, we talked about some of the differences between those two kinds of animals. And then from there, we'd start looking at some more specific features that would maybe allow us to classify this new animal with an actual phylum. Maybe it's an annelid, or maybe it's a tardigrade, or whatever. It's any other of the many groups of animals beyond just sponges and cnidarians once we know some of those other features. Now, the more contemporary, modern way of looking at things is based a lot less on what animals look like and a lot more on molecular evidence. So let me just take a quick moment to talk about what that means. Traditionally, when people classified animals, they just did so based on physical appearance. And so if two animals were kind of sort of similar looking, they were placed into a similar category. Maybe they're in the same family, or maybe they're in the same class, or maybe in the same genus, right? But it turns out that sometimes animals look alike not because they're related, but because maybe they evolved in similar habitats with similar environmental challenges, and so maybe they came up with some of the same types of adaptations, not because they're related, but because of natural selection in their own respective environments. In that case, if you put the two animals together in the same group, you'd actually be mistaken, and you'd create a taxonomy that was incorrect and flawed, which sometimes happens. With molecular data, what we look at is percent similarity of DNA sequences or percent similarity of protein sequences. And so those kinds of things are not subjective. They're very objective, actually. They're very much based on numbers. You compare the number of A's and C's and G's and T's in the DNA, and if two organisms are 99% similar, they're 99% similar. And if those two are 99% similar, and there's a third one that's only 97% similar to the first one, then you know that guy's a little bit more of a distant cousin. Right? So it's a lot less open to interpretation and a lot more based on sort of cold hard numbers, which makes it a lot more, makes us have a lot more faith in the results that we see in that kind of data. All right, so this is an example of that, and this is actually a fairly small group of animals. The Mysostomids are an example of a group that initially was classified as being very, very different from what it actually is now. Initially, they were classified as being part of the annelids because they look kind of superficially like an annelid. They look sort of like a segmented worm, like an earthworm or a leech or something like that. But it turns out that segmentation is not because the Mysostomids are related to annelids, but because of convergent evolution, where they evolve in similar habitats and therefore can come up with some of the same features. So by looking at the genetic analysis of these Mysostomids, they've actually been reclassified now as not being part of the annelids, but actually being more classified or more similar to a flatworm, which is a very, very different kind of animal than what an annelid is. Much simpler, very different lifestyles, very different ecologies, very different ecological roles. This is all based on genetic evidence. It's all based on molecular evidence as opposed to just, some crusty old taxonomist sitting in a basement of a museum trying to figure out what two things look like. This is actually based on DNA sequences of these organisms, which make them more easily classified in a more accurate manner. So this is the idea that we're dealing with now. Morphological features, physical traits, those kinds of things are less favored now. And genetic differences, even entire genomes. We can actually sequence an entire genome, so all the genes an organism has. We compare genomes among different organisms to see who's related to whom, and we kind of build backwards and make a family tree from that. So it's a lot more concrete because, like I said before, it's based on numbers. So the metazoans now are broken up into two different categories. This is, again, a more contemporary approach to classifying the animals, but the first separation that's kind of different from the old traditional approach to looking at animals is that the annelids and arthropods are kind of separated into two distinct groups, almost like two teams of metazoans. And so within the annelid group and the arthropod group, we then take a look, basically both of these are protostomes, right? So we take the protostomes and we divide these up into the ecdysozoans and the spiralians. And really the main difference there is ecdysozoans are animals that go through a process called ecdysis, where they shed their outer cuticle, or kind of like their outer exoskeleton sort of a structure, and they grow a new one. So ecdysozoans would include things, most remarkably things like the arthropods and the nematodes, which are the roundworms, because they are both animals that definitely go through ecdysis. Spiralia would be things more like the mollusks, would be a really big group of Spiralia. So that's another feature that we would take a look at as well, with a newer approach to classifying animals. And then within the Spiralia, it gets a little more complicated. Lophotrochozoa, Platyzoa, those are kind of subdivisions of the Spiralia, but that's getting pretty specific and not really a level that I typically go into too much in these intro-level classes. So this is the entire family tree of the animals looking at the modern day approach. I know that even in my majors biology classes, I don't definitely go through all of these. Typically the groups we cover are the sponges. We'll talk about what choanoflagellates are here in a second, but sponges, Ctenophora, Cnidaria. We'll talk about the Acela a little bit. Rotifers, mentioned Cycliophora, flatworms. Let's see, annelids, mollusks, Nemertea, nematodes, definitely those. I mentioned tardigrades a second ago. These are water bears, really cute little animals. Don't talk about them a whole lot, but they've got some really unique things they do. They're actually able to go into suspended animation for years or decades or probably centuries. They can even survive in space because they go to this resting stage where they're absolutely resistant to the environment and they can survive really all kinds of nasty conditions and then reanimate. And they actually look like little tiny microscopic bears, which are really cute. There are arthropods. We talked about those before as well. Echinoderms. And then these last three groups are the chordates. We are vertebrates, of course, as humans. So, in majors biology, we go through maybe half of these at most. If you're taking more of like an intro biology course, then it's, we'll mention a few of these maybe, but it's really not going to be a whole lot of depth with any of these groups. You can literally take entire classes just on basically this slide. It's a huge, huge amount of information there. Now, the other thing that we talk about in this chapter is where we think animals came from. In other words, what other group of organisms produced the animals? And so there's limited options, right? You could be bacteria, archaeobacteria, plants, fungi, or protists. Those are basically the other 5 kingdoms that could have given rise to the animals. So we know which one it was. We know which group it was. It was the protists that gave rise to the animals because of molecular similarities. There's no doubt that animals are more related to protists than they are to fungi or plants or bacteria or archaea. But the question is, how did that happen? All right, so there essentially were 3 hypotheses that tried to explain how animals might have arisen from single-celled protists. And so, out of order here for some reason, the first one was a multinucleate hypothesis. And essentially what this said was that maybe there was a cell that for some reason began to do cell division, so it did mitosis, but never completed cytokinesis. And it just kept doing that over and over and over again, so you ended up with this one like supercell that had maybe hundreds or even thousands of nuclei in this one single cell, and maybe all those nuclei, instead of functioning as nuclei, maybe each one of them began to function as an individual cell. So this one cell with 100 nuclei actually became 100 cells somehow. That hypothesis is the second best one. It's really not very likely. It's probably not even close to being the best one, but it's not as bad as the worst one of these 3. So it's, not a bad idea, but not really quite what we think actually happened. The second one, the colonial flagellate hypothesis, takes a group of organisms like this over here. So this is actually a mass of cells called choanoflagellates. We mentioned them before on this previous slide. Choanoflagellates are all the way over here. They're a cousin group to the animals. They're an offshoot, or they're part of, actually, the protists. And these choanoflagellates are, like I said before, colonial. And colonial, but what we mean by that is they are made up of large aggregations of cells that are kind of living together, but in the protist sense, each cell is kind of doing its own thing. So they're not cooperating. Just because they're together in one mass doesn't mean that they're specializing or forming tissues or doing different jobs, but they are together in one mass. Now, what you could imagine happening is that maybe some of those cells in that mass began to take on just a little bit more of a preference for one kind of a job, and other cells begin to take on a preference for another kind of a job. Right? And so eventually these cells that were beginning to become specialized got so specialized that they relied on the other cells in the mass for their own survival. And every cell in the mass began to rely on everybody else for their own survival. So that could have produced something like a multicellular animal, and that's exactly what we think happened. All right, now there are some protists, not necessarily choanoflagellates, but there are other protists that are observed to have a degree of sort of a colonial aspect to more of a multicellular aspect, tissue level aspect. And there's a whole gradient in between from cells that just hang out together, but they actually are exclusively functioning independently of each other. There are some that cooperate a little bit. There are some that cooperate a lot. There are some that are full-blown multicellular tissue-forming types of things. So it's not all that far of a reach to think that that's how the animals actually evolved, was from a group, probably a single species of choanoflagellates that eventually got to be so cooperative that they couldn't survive without each other. And at that point, we would say that they are multicellular. Okay. Now, the third hypothesis, the polyphyletic origin hypothesis, we're not going to say too much about that one because this one's actually the least likely to hold any water at all. The polyphyletic origin hypothesis says that basically there were multiple groups of organisms, many species that all became animals kind of at the same point in history somehow, simultaneously and coincidentally and randomly. They all became animals at the same time, which, as you can imagine, is probably not the most realistic hypothesis. It's actually the least favored of these three. The second hypothesis is the one that definitely is most agreed upon by taxonomists as far as where animals came from, colonial flagellates. Now, another aspect of the evolution of animals that we should talk about just really briefly is called the Cambrian explosion. Now, first off, the Cambrian explosion is not literally an explosion, like it's not something blowing up. The Cambrian explosion refers to an explosion of diversity. In other words, a whole bunch of species showed up at around the same time. We went from having hardly any animals to having 30-some phyla of animals over the span of only a few million years. Now, a few million years in human terms is a long time, but in evolutionary terms, a million years is a very short time. So the fact that so many animal groups popped up at around the same time over the span of only a few million years sort of raises a question, like, why did this happen? Like, what caused this massive explosion of diversity where we go from having hardly any animals to having all these very diverse and amazing different types of animals that we see today, and more that have since gone extinct. And so it turns out that there was, we think, the development of a group of genes called Hox genes, or homeobox genes. So Hox genes are regulatory genes that get to control when other genes are turned on or turned off. Right? So it's like a master switch. It's like on a circuit breaker, you've got all your individual switches for different rooms in your house, and then you've got one master switch that shuts them all off or turns them all on. All right, the Hox genes are kind of like the master switch. They decide, maybe not every gene in the genome, but maybe one Hox gene might control how 100 other genes are expressed, that kind of a thing. And so you can imagine that if you all of a sudden had a mutation in that one master switch gene, what would that do to the body form of the animal? It would change it immensely, right? So if you change how that one master switch gene works, all the other genes that it controls are going to be changed in their own expression as well. So the development of these Hox genes produces the possibility of a mutation occurring, which could then in turn affect how all of these other genes are going to be expressed. And that, we think, is how animals got to be as diverse as they are today. So with that, we're going to kind of wrap up and get this posted to YouTube. And of course, if you have any questions, you can do my best to try to answer those questions for you.